It is my pleasure today to introduce Dr. Shelly Flagel. She's Associate Professor in Psychiatry at the University of Michigan, also the co-director of the Michigan Neuroscience Institute. Dr. Flagel, thank you for being here today.
Thanks so much for having me and for doing these in general. Really appreciate it.
Of course, my great pleasure to talk to such scientists such as yourself. So I guess we'll start the conversation with a recent paper from this year in psychopharmacology investigating individual differences in opioid taking and opioid seeking behavior in male rats. Can you tell us a little bit about the main findings of this paper?
Yeah, so we are interested in assessing what individual differences might predict opioid taking and opioid seeking behavior. And so we were looking at this from the perspective of sign and goal trackers and high responders and low responders. So for those that don't know, there's a rich history in studying individual differences in the field of addiction. The high responder, low responder model was first put forth by Piazza in the 80s. And essentially what that is, or what that showed, was that if you place rats into a chamber, a novel chamber, those that have great locomotor activity will be called high responders, and those with less locomotor activity will be called low responders. And essentially what he went on to show was that the high responders are those that acquire drug-taking behavior more readily than the low responders. And so this was initially shown with psychostimulants and then expanded to things like nicotine and morphine. And in contrast, the sign and goal tracker model, for that we capture individual differences in how rats respond to Pavlovian reward-associated cues. So you put them into a chamber where they receive repeated presentations, in this case of an illuminated lever paired with a food reward and adjacent food cup. So it's classical Pavlovian conditioning, no response is required, yet when we do this task, we see that there's this great individual variation. So on the one extreme, we have what we call sign trackers who become attracted to the cue, in this case an illuminated lever. They're attracted to it, they interact with it, grasp it, gnaw on it, chew on it, almost as if it were the reward itself. The cue itself becomes desirable and an attractive stimulus. And on the other extreme, we have goal trackers. And goal trackers see the cue essentially for what it is, that is a predictor, they'll orient towards it, and then they immediately go to the food cup and wait for food to be delivered. And so we and others have shown that this sign tracker, goal tracker model isn't necessarily a great predictor of the initial acquisition of drug-taking behavior, but more so a very good predictor of relapse propensity, and in particular, cue-induced relapse propensity. And if you think about that, that makes sense. So in individuals with addiction, one of the biggest problems of the treatment of addiction is relapse, and individuals will report relapsing when they come into contact with people or places or paraphernalia associated with the drug-taking experience. And so this process that we're studying in sign and goal trackers, and in particular in sign trackers, that is that process of these reward-associated cues to gain inordinate control over behavior, we think is akin to that same process that leads to cue-induced relapse in human drug-seeking behavior as well in addiction. So the point of this paper was to say, while we've done a lot of studies with psychostimulants using the sign and goal tracker model, there hasn't been much done with opioids. There has been one study by Lindsay Yeager and Terry Robinson where they looked at Pavlovian conditioned cues associated with opioid infusion, but not necessarily instrumental drug-taking behavior. And so that was one of our initial prompts for doing this, was to determine whether or not sign and goal trackers differ in opioid-seeking behavior following opioid self-administration. We used remifentanil in this case, in part because if we want to get at that notion of that cue reward pairing, remifentanil is much better suited for that because of its fast-acting properties. So we can show an illuminated light, in conjunction with delivery of the drug, and it will be tightly paired. But that may not be the same case for other opioid drugs. So that was one reason we used remifentanil, and then nobody had really assessed, to my knowledge, individual differences with the high and low responder model with respect to remifentanil. And we also have begun sort of repeatedly with various studies, we often will measure both sign and goal tracking tendencies as well as high and low responder tendencies to see if we can determine whether one trait may be more predictive of a certain aspect of addiction-related behavior than others. And that's essentially what we did here. So the main findings were that neither the propensity to sign track or the propensity to be a high responder, that is a sensation-seeking behavior, seemed to be a good predictor in our experiment of either the initial drug taking of remifentanil or the drug-seeking behavior, that is relapse behavior, reinstatement following the drug-taking experience.
Wow, so this is really a major difference then between this opioid drug and psychostimulant drug you mentioned before, cocaine. That's right?
Right, correct. So yeah, and so then that prompts a number of questions, right? So one, of course, is the caveat that, people aren't necessarily abusing remifentanil on the streets, and is it a good model of, opioid addiction or opioid addiction tendencies? That's one question. But assuming that it is, and assuming that we are really capturing, different predictors of opioid taking and seeking versus cocaine taking and seeking, I think that's really important to know. And I think it's a point that's often lost. We try to come up with the all-encompassing theory of addiction, whether it's the dark side of addiction or the incentive sensitization theory of addiction. But in reality, I think we all know from reading anecdotes in humans that it's not the case. There's going to be many very different triggers of addiction depending on the drug of abuse, depending on a number of environmental circumstances, depending on genetics. And so I think we need to get over this notion that there's an all-encompassing theory of addiction that's going to apply to every drug and every individual. And we need to not just embrace individual differences, but embrace different drug classes too. And note that, maybe it's a case, something I've always thought about is maybe the dark side of addiction is particularly relevant for drugs like opioids, whereas incentive sensitization is particularly relevant for psychostimulants. There's still more to be determined, but I don't think they necessarily are all mutually exclusive, and I don't think we should study them as such.
Oh yeah, I would totally agree. And it also, another thing you mentioned was that kind of caught my attention was you chose remifentanil because it's such a fast-acting opioid, and the Q pairings can be made, one after another, essentially, because the drug wears off so fast. Unlike, for example, heroin, which is a longer-lasting opioid, and they take it less frequently. I guess would be part of the implication of that. And I'm curious, there's also like long-lasting stimulants, like methamphetamine, for example. And I'm wondering if that might be a sort of a little bit of an entree into dissociating these factors in terms of how this works in the brain and relative relevant sensitive sensitization and that kind of thing. My impression from doing a little bit of this kind of research is that, like, for example, Q reinstatement in our typical model that we both use is like really pretty massive for meth and not so much for heroin. In our hands, pretty high for remifentanil too, actually. I'm just curious if you have thoughts about like how those factors interact with one another.
Yeah, no, that's a great point. And it also reminds me to note that our levels of reinstatement, while they were actually relatively low for us. It was definitely a difference in terms of the Q-induced reinstatement compared to extinction levels, but lower than what we might see for other drugs. And so it's something that we've thought about as well. And by all means, we know that kinetics plays a huge role, and not just kinetics of the drug, but in terms of the rate of infusion. So work from Anas Maha and Terry Robinson and others have done a great job highlighting that as another really important factor. And so that's something else to think about in not just modeling addiction, but how our experiments are designed and how those types of things, when you think about the replication crisis and how we interpret things, like those things, like how fast the animal is receiving the infusion could play a really important role. And there's no doubt that the pharmacokinetics of the drug itself, once it's in, is also a big factor.
Absolutely, yeah. And another question I had that kind of relates to the issue of individual differences is, this particular study is in male rats. Do you have anything sort of to say about how these factors interact with sex as a variable?
Yeah, another great point. And so this is definitely something we're beginning to assess. Unfortunately, and I'm almost embarrassed to say, all of our studies initially with cyan and gold tracking were done with male rats. We've done, in terms of the ones that have looked at addiction-like tendencies. And so that is top on our list. We have to figure out if there's actually differences in addiction-related behaviors in male and female cyan versus gold trackers. And even high and low responders, we haven't done a good job of that either. And so while in terms of the cyan and gold tracking cyan and gold tracker population, it's really interesting in terms of the sex differences. We've done this with very large populations because these were part of genetic studies. So talking 2,000 rats essentially, and comparing males and females. And if you were to take just the raw values of male cyan trackers to female cyan trackers, you actually wouldn't see a difference. But if you look at the population of a whole and characterize them as cyan and gold trackers, based on those last few sessions of Pavlovian conditioning, you see that the female population is skewed more heavily towards cyan trackers than the male population. And essentially what's driving that, interestingly, is the intermediate population is actually not so much an intermediate population in females, but sort of much more skewed towards cyan trackers. So there's less of an intermediate population in females. And so there's much to be investigated there. And it may very well be the case that, I don't know if we would expect that females will also show, female cyan trackers will also show enhanced, for example, Q-induced reinstatement compared to male cyan trackers, or maybe it will be that they actually show differences in acquisition that we didn't see in male cyan versus gold trackers. So it's definitely an interesting area that we need further investigation.
Wow, yeah, that's definitely, it sounds like a bunch of very interesting, important questions that still remain to be seen, even after all the work you've already done in this area. And just as a small anecdote, many years ago, I think around the time probably when we first met, this would have been when I was a graduate student at Michigan, we were starting to do some sign and goal tracking experiments as well. And we were very sort of, in Kent Berridge's lab, we had a much smaller scale than the Robinson group did in terms of operant box space and stuff like that. And so every rat was very, very precious to me, a graduate student. And so I ended up actually doing those initial studies only in females, based on the observation that we wanted them to acquire the sign tracking as fast as possible. And in our hands, it's limited N and everything like that, obviously compared to what you guys have done over the years, that was the females. And so we were it also was convenient for me because most of the rest of the lab, as almost the rest of neuroscience, was using only males. And so there was breeding colony, a bunch of females were around, and those were using mine. Cool. So let me ask you also then, you've been doing this sign and goal tracking work for many years now. Can you tell me a little bit about how your thoughts about how that individual difference between sign and goal tracking, or perhaps this intermediate behavior you mentioned, which I understand would be them kind of going back and forth between the two cues during the cue presentation. How is your kind of conceptualization of that, or your interpretation of those findings and their relevance to addiction evolved?
Yeah, great question. So, initially, like I alluded to before, we characterize rats based on their tendency to approach the cue that's predictive of reward. We found that after a number of tests and a bunch of studies that a number of groups done, it was pretty obvious that sign trackers are attributing this excessive incentive motivational value to the cue, as well as predictive value, whereas goal trackers are only attributing predictive value. And so that then allowed us to do a number of great studies, not just from my group, but others as well, to kind of dissociate the neural mechanisms that are underlying predictive versus what we call predictive versus incentive learning. So, of course, this stems from the incentive sensitization theory and the notion that, well, if they're attributing this excessive incentive motivational value to the cue, then they must be more susceptible to incentive sensitization following drugs of abuse. And a number of our early studies suggested that was the case. So sign trackers show enhanced psychomotor sensitization relative to goal trackers. There's a lot of great work from Ben Saunders showing that they have, enhanced attribution of incentive value to drug-associated cues as well. But then you fast forward a number of years and two things emerged. One was that it wasn't just that goal trackers or that sign trackers were, showed increased evidence of cue-induced reinstatement or relapse behavior. But interestingly, for goal trackers, they were more susceptible to context-induced reinstatement. So that was the first kind of light bulb moment where, okay, well, let's rethink this. And it's not just, sign tracking bad, goal tracking good. There's different aspects to this, and it's definitely not black and white like all of science. And so that was kind of the first hint that there's much more to this than what we think. And so goal trackers really interestingly not just use contextual regulation of their drug-associated cues, but work by Jonathan Morrow showed that they also use context to more appropriately mediate the response to fearful stimuli or aversive stimuli. So then that led to the notion that sign trackers are sort of a better model of post-traumatic stress disorder like behavior, in the sense that they can't properly utilize contextual cues to regulate their fear response. And so that was sort of one aspect, that the sort of context versus discrete cue and how they use this contextual regulation. And then you fast forward, and Alex Kawa from Terry Robinson's group did this sort of prolonged intermittent self-administration and showed that after prolonged intermittent access to drugs, you actually don't see differences later on in addiction-like behavior between sign and goal trackers. And so that was the other big hint that it's not as simple as just, pure incentive sensitization theory and sign trackers susceptible to addiction, goal trackers not. It's much more complex than that. So the way I'm thinking about it these days is that, but having said that, if you want to study cue-induced relapse following, either short or long periods of abstinence and capture that vulnerability, then the sign tracker goal tracker model is perfectly appropriate for that. And so there's certain aspects that if you want to capture these individual differences, it makes perfect sense. But how I'm thinking about it these days is more as we're, just simply characterizing the rats based on Pavlovian conditioned approach behavior. We're able to capture this really complex neurobehavioral endophenotype that's not just of relevance to addiction or certain aspects thereof, but it's of relevance to a number of psychiatric conditions. And I actually am hesitant to say psychiatric conditions, but I would say psychiatric symptomatology. So we're capturing these symptoms that are evident across psychiatric disorders. So sign trackers are, they show attentional deficits, work by Martin Sarter's group. I already talked about the PTSD-like behavior by Jonathan Morrow's group. Myself and Vedran Lovics showed that they differ in impulsive behavior, so sign trackers are more impulsive than goal trackers. And remarkably, they have these unbelievable differences in their brain that make perfect sense for all of these things to be happening. And we still have a lot more to work out in terms of the brain mechanisms. But like I said, I think it's very clear we have this neurobehavioral endophenotype of relevance to psychiatric conditions that can teach us a lot.
Wow, that's fascinating. The sort of abnormal context responding that you mentioned in, I guess, depending on how you look at it, either sign or goal trackers, right?
Right.
Like, that really strikes me as very fascinating. And have you guys started looking at, when I think of contextual regulation of drug seeking, I think of hippocampus. Have you noticed anything unusual there, or anything that might be explanatory?
Right. So Jonathan Morrow's group is starting to look into that circuitry, and has definitely just had some really interesting data in that regard. And so they're the only ones focusing on that right now that I know of, that I'm aware of. We've stayed away from the hippocampus. We're thinking of it more in terms of goal trackers using this contextual regulation, but also that's part of this sort of cortical cognitive control strategy that I think they rely on a lot of different decision-making aspects. And so we're thinking it more of kind of a cortically driven response and dopamine independent. And that goes back to the really old literature that kind of showed these types of contextual regulation would be both dopamine independent and cortically driven. And specifically, a lot of our work in recent years has surrounded the paraventricular nucleus of the thalamus as kind of a central hub that may be mediating this neurobehavioral phenotype of goal trackers versus sign trackers. So my thought is more that goal trackers are receiving this top-down cortical control from the cortex to, say, this midline thalamic nucleus. And while sign trackers may have that in place, they also have this kind of subcortical overdrive, this enhanced motivational and arousal systems that are overriding any cortical control coming in. And of course, that's just one part of the brain, or one circuit or network, that I think is really important. And I'm sure the hippocampus is equally important, especially when you think of learning and memory and context. But I'll leave that up to Jonathan Morrow to figure out, and I'll focus on the PVT and surrounding circuitry for now.
Right. Very interesting. So am I sort of interpreting it correctly, what you just said, that this is, are you thinking of this as kind of like top down versus bottom up control of behavior more generally, or is that too simplistic?
No, it's not too simplistic at all. That's exactly how I'm thinking about it. And I can tell you, so the specific circuits, like corticothalamic striatal versus hypothalamic thalamic striatal, and kind of this battle where I think it's the PVT that has this sort of, it's acting as fulcrum in this regard. And when those cortical, again, going back to psychiatric symptomatology, when you have an imbalance between that cortical versus subcortical input, that's when you get these maladaptive behaviors that may be characteristic of psychiatric symptoms.
Okay, so you mentioned that the PVT seems to play a role in mediating these top-down versus bottom-up influences. Can you elaborate a little bit about what kind of role you think it's playing in those processes?
Yeah, well, it's of course to be determined, but the questions that I have surrounding that are whether, are, for example, sign and goal trackers wired differently, or is that the PVT is acting differently in terms of how it integrates the behavior coming into it and or how it sends it out of it. So the circuitry in particular that we've shown to be involved surrounding the PVT in sign versus goal tracking behavior happens to be, so what's interesting is that in terms of sort of being wired or encoded differently, is if you look at neurons in the prelimbic cortex that are projecting to the PVT, and if you look at cue-induced neuronal activation in those neurons, it's actually not different between sign and goal trackers. So that tells you that the predictive encoding of the cue is likely happening within that circuit. That seems to be the same between the phenotypes. So then something maybe subcortically may be driving the sign tracking behavioral phenotype. And our evidence so far suggests that specifically neurons from the lateral hypothalamus of the PVT are sort of hyper-regulated, hyperactive in sign trackers compared to goal trackers. And also, I think, and perhaps most importantly, is that final common pathway from the PVT to the ventral striatum, in particular the nucleus accumbens shell. those neurons are activated to a greater extent in response to the cue in sign trackers compared to goal trackers. So there seems to be this incentive motivational encoding circuit that's pretty well worked out from our prior studies in terms of just cue-induced CFOS activity. And now we have this kind of blueprint to work around to see how we can use different techniques like chemo and epigenetics to play with that weighing of the different circuitry and to determine what that neural code is in the PVT and whether or not it maps onto behavior.
Really interesting. And then with regard to the PVT, just one last question. You know, one of the other things that I understand that people think it may be involved in is kind of mediating sort of conflicting motivations as well, like kind of appetitive versus aversive motivation, that kind of thing. How do you think this fits into, like the story you're telling us about, fits into that conceptualization, if at all, or if you know?
Yeah, no, fantastic question. There's lots of great work coming out in that regard in terms of avoidance versus approach behavior and conflicting decision making. So work from Gavin McNally and Fabrizio De Monte, like just amazing work in that regard surrounding the PVT. And I definitely think it's relevant. You know, I think it kind of forces us to take a bigger step back and what are these sign and goal trackers really teaching us, and think about it more like, you know, it has to do with a lot of motivational processes and decision making and maladaptive versus adaptive decision making. And so I think it's very relevant and makes perfect sense in terms of you know, the more aversive and conflict types of decisions in terms of sign versus goal tracking. Not much has been done in that regard. So as I alluded to before, Jonathan Morrow is looking at aversive in response to aversive related stimuli. But there's really not much. I would love to see the differences in sign versus goal trackers in terms of conflict decision making and how they respond and what's going on in their brains at the same time.
Wow, super interesting. As usual, there's plenty more work for us all to be doing in this domain.
That's right.
I'm really looking forward to seeing what else you guys discover in the next coming years as well.
Thanks. Thank you.
Yeah, well, it was really a great pleasure to interview you, Dr. Flagel, and thank you so much for your time. I really appreciate it.
Thank you. My pleasure.